We're going to now cover one of the most important methods that we can use for human subject research, which are randomized controlled trials. This is going to be a lightning fast intro that's meant to give you the basics and a set of practical tips to running RCTs. You can take a full course or many courses that cover RCTs, but we'll cover what we can in 10 minutes. And I highly encourage you to do further reading. This talk is geared towards folks that haven't run an RCT before, so we want to give a motivation for what they are, why they work, and some practical tips for implementing them. So in order to understand RCTs, we're going to work through a case study together. So in this case, there's a really interesting paper by Noy and Zhang, where they were interested in the productivity effects of ChatGPT in the context of mid-level professional writing tasks. So like all good research, they start with a really strong and interesting research question. And what makes this a good research question? Well, the first is that they have what we call preordained outcome measures. So they've told us sort of upfront, what is the phenomenon that they're interested in measuring? In this case, the productivity effects of ChatGPT. Now, they haven't gotten specific about that yet, but we're going to talk about that later. And for a research question, this is a great place to start. The second is that they've given us a specific context. So it's not just the productivity effects of ChatGPT in general. They tell us on mid-level professional writing tasks. And what this means is that we have, a sort of specific context, a specific set of tasks we're interested in, and usually with that, sort of a population that we may consider studying. And so while this research question is sort of succinct, it's specific enough that it gives us a starting point for maybe designing an experiment. And so we're going to cover today the three elements of an RCT. There are many more than three elements, but these are sort of big ones. And so I think they'll be helpful for understanding RCTs and potentially running one. So the first is randomization. The second is preordained outcome measures, which we talked about briefly before, and blinding. So first, why can't we just observe people that use ChatGPT and those who don't? So concretely, what we could do is send out a survey to MIT students, ask them to participate in the study, and then we simply measure the writing productivity of those who do use ChatGPT and those who don't, and compare the two groups, right? There's two problems with this. The first is called selection bias. So this is basically when the population that you actually care about is different from the sort of sample that you use and that you run your experiment on. So in this case, let's say our sample is MIT students, but the population we really care about is all professionals that are using ChatGPT or have the potential to use ChatGPT. There's probably a somewhat of a difference, right, between MIT students and people out there in the real world using ChatGPT or potentially using ChatGPT for their work. And so the effect maybe that we detect for MIT students might be systematically different than the effect that would happen when we actually, let's say, introduce ChatGPT into the workforce. So that sort of selection bias can be a problem depending on like how you actually recruit the participants into your study. The second is confounding. And so we're going to actually go through an example of confounding on the next slide. So what can we do? So let's say we have an initial sample of MIT students. There's some of those that are more experienced writers than others. So in this case, the blue dots are experienced writers and the green dots are less experienced writers. So we just let people go. Basically, some of them will use ChatGPT and some of them won't. And maybe what happens is that people that are more experienced writers are actually less likely to use ChatGPT because maybe they, are just better at writing, so they don't feel that they don't need assistance. They maybe like their own writing style. And versus the people that are less experienced might actually sort of choose to use ChatGPT more because it helps them. They feel like it can give them a larger boost. And so the problem with this, what we call selection into treatment, is that now when we go to measure these two populations and their productivity of writing, maybe the thing we're actually capturing is not the effect of ChatGPT, but this sort of selection into treatment where the people that use ChatGPT are actually less experienced writers, and the people that don't are more experienced writers. And so whatever sort of effect of ChatGPT we might detect is being washed out by this selection into treatment problem, where it just happens to be that people that are better at writing are tending to not use ChatGPT, and people that are not as experienced as writing are using it. And so this is not what we want. So what's a better way of doing this? Well, now given our initial sample, instead of just letting people choose whether they want to use or not use ChatGPT, we actually force people to either use or not use ChatGPT to do a certain writing task. And then in this world, ideally what you see is that in the green rectangle and the blue rectangle, you have equal or roughly equal amounts of sort of experienced and less experienced writers. This is a very idealized case. And so obviously you won't always end up perfectly balanced on all of your confounders. But that's the idea with randomized control trials, is that you're sort of mitigating this, what we call selection into treatment problem, where, for example, students that are better at writing choose to not use ChatGPT and vice versa. So this is what we want. The next part of an RCT that we really want to think hard about is the outcome measures. So the high level takeaway for this should be decide what you want to measure and be concrete about it before you run your RCT. In this case, it might be number of tasks completed in a certain period of time. It might be the quality of writing as determined by human raters. It might be the factual correctness of answers when using ChatGPT. And so just some quick tips about your outcome measures. Try and be precise and exhaustive on your data collection and analysis before you run your experiments. So as a rule of thumb, the more sort of planning you do in terms of the data you collect and the analysis you want to run, the better off you'll be. The experiments can often be quite expensive to run. And so the better you have it planned out from the get-go, the better off you'll be. And so some quick tips on like what other data you might want to collect besides just your outcome. Demographics. So and that is often a factor that affects your outcomes. And then also think hard about your own specific research question. So in this case, writing experience might be a big confounder, right? And so we want to make sure our groups are balanced from on that dimension. And if you collect this data up front, you'll sort of be able to do a post-hoc analysis where you can balance the data. Versus if you never collect this data in the first place, it's going to be very hard after you've collected it to sort of figure out if there's any confounding going on. And then the final thing is that a lot of experiments don't work, which is totally fine. We learn a lot still from experiments that don't work, and sometimes a surprising result is really interesting. And so, yeah, as long as you have a strong analysis plan up front, you have an interesting research question. If the experiment doesn't go as expected, that's totally fine and that's still valuable. Okay, now we'll move on to blinding. So we'll go through this very quickly. So the way that you sort of understand blinding is who knows what about the experiment. And so there's two main ways of blinding. The first is single blind, where only the researcher knows who got the treatment. So an example of this might be, surgery, where the surgeon knows, what surgery is being given to which patient probably, versus a double blind experiment, which is probably what you'll be doing, which is where neither the researcher nor the subject knows who got what treatment. And this is sort of the ideal case where you have the least chance of bias, because as a researcher or as a subject, you'll be less prone to sort of act in a way according to the treatment that you got, right? So finally, we want to cover some practical analysis tips for when you go and do your RCTs. So the first bucket is what we call sanity checks. So make sure you include what we attention checks in your study. So the main point of this is to make sure your participants were actually doing what you asked. A lot of people will run online surveys, and it's very easy for people to just skip through those and do them quickly without really trying to engage with what you want them to engage with. So make sure you include these attention checks to ensure your participants are doing what you asked. The second is to ensure proper randomization. So look at your treatment and control groups, see that they're balanced on the things that you expect them to be balanced on, which should be most sort of demographics, experience, for example, opinions. They should be roughly balanced on those. And then finally, make sure you try to understand whether your subjects actually complied with the treatment. So if we take the case of the ChatGPT as a writing tool, it might be bad or interesting if you gave people ChatGPT, they were in the treatment condition, but they didn't actually use they didn't use ChatGPT to assist them. So in that sense, they weren't quite complying with the treatment, and so you're not really capturing the effect that you want to capture, which is if you did actually use ChatGPT, would that help you or not? And so it would be good to measure basically to what extent did your participants actually comply with the treatment that you assigned them. And then after you've gotten your results, the next question is like, what do I do first? The very first thing you can do that's very simple is just plot the difference between your treatment control outcomes and try to have a sense for what a big deal is or isn't. So don't rely just on p-values. Again, let's take the productivity effects case as an example. If let's say on average the tasks take 10 minutes, and then the effect size we see is 2 seconds. That might be statistically significant depending on how many people you had in your study, but you sort of have an intuition that okay, 2 seconds is not like a huge deal. And so it's important to have a sort of notion of like what is a big effect and what isn't, so you can properly frame your results. A lot of studies will have statistically significant outcomes, but sort of fail to really think about like, okay, what is the effect size? Is this a big deal? Is this not a big deal? That kind of thing. The second practical tip is to plot the distribution of outcomes. So I've run several studies where participants are supposed to answer on a scale of 1 to 7, and a lot of people are just giving answers. Almost everyone is giving either a 1 or a 7, and so this might tell you something about what the phenomenon you're studying, maybe the way you're posing the question, and it can often prevent you from being fooled by averages. And so you can it's really important to sort of understand the distribution of responses along instead of just looking at basically, okay, what is the average here, what is the average there, and comparing them. You could use regressions to control for any confounders not balanced by randomization. This sort of ties back to what I was saying about trying to be exhaustive with your data collection and making sure you sort of collect any potential confounders up front so that you can sort of balance them in your post-hoc analysis. And then finally, it's important to note that qualitative analysis can be super helpful. So if you collect comments from survey participants, it can be very insightful. So you can sort of get ideas for hypotheses for the mechanism of like why your treatment worked or didn't work, especially in the case where it didn't work. Oftentimes participants might give you some idea of like why that may be. And so it's really good to just give free-form answers as well, even if the sort of metrics you're collecting are quantitative. And so here's an example basically of the first plot you might make. So these are two outcomes that we are interested in. You can see in the first case, we have in both cases, we have three conditions: the controlled treatment, the control, the standard, and the treatment conditions. In the first outcome, we don't see any difference between any of the conditions. While in the second one, in the treatment condition, we do see a difference. And these are scales of 1 to 7, I believe. And so this might be like the first plot you make after running an RCT. So that was like a super fast introduction to RCTs for those that haven't run it before or aren't familiar with it. There's so much work. They're one of the most powerful methods for human subject research and beyond in almost all the sciences. And so hopefully this was helpful as a quick primer. And I highly encourage you to look at other resources because this is a really, really powerful technique and important to understand. So thanks.